It's Friday, 8:30. This is the day when we're celebrating vision research, and today is no exception. So we have our guest speaker from Massachusetts. We're very delighted. And with that, I would like to ask Alex to introduce our speaker.
Good morning, everyone. My name is Alex Turak from Dr. Palczewski Lab, and it's my distinct pleasure to introduce our today's speaker, Dr. Janey Wiggs. Dr. Wiggs is Paul Austin Chandler Professor of Ophthalmology and Vice Chair for Clinical Research in Ophthalmology at the Harvard Medical School. She also serves as a clinical associate at the Mass Eye and Ear Hospital in Boston, Massachusetts. Dr. Wiggs is an accomplished clinician scientist specializing in the genetics of glaucoma. She acquired her PhD in biochemistry in 1981 from UC Berkeley, followed by the medical doctor degree from Harvard Medical School in 1985. She completed her residency in the Harvard Ophthalmology Residency Training Program, followed by fellowships in glaucoma at Mass Eye and Ear, as well as fellowships in medical genetics at both Mass Eye and Ear and the Tufts University of Medicine. In her work, Dr. Wiggs aims to identify genetic factors that underlie various forms of glaucoma, including adult onset primary open-angle glaucoma, pseudoexfoliation glaucoma, juvenile open-angle glaucoma, and others. She's a co-author of over 250 peer-reviewed publications in many prominent journals in the field, as well as 22 book chapters. Her research, which has been continuously funded by the NEI for over 20 years, has provided critical information regarding the biology of glaucoma and contributed to the improvements in diagnostics and treatment of the disease. Dr. Wiggs is recipient of numerous academic and teaching awards, including most recently David Epstein Award in 2018 and the Mildred Weisenfeld Award in 2020, both from ARVO. She was also elected to the National Academy of Medicine in 2018. And the title of today's talk is Using Genetics for Glaucoma Risk Stratification. So without further ado, I'd like to welcome our today's speaker, Dr. Janey Wiggs.
Great, thank you so much. Thank you very much for that nice introduction, and thanks very much also for the opportunity to speak to you all today. I hope you can see the slides and also hear me, because with these virtual arrangements, you can never be too sure. I'm also, excited that it's a virtual talk, but in some ways I kind of wish that I was actually there in person, given the weather here today in the northeastern United States. So today I want to talk to you about some really exciting work that's been going on in genetics of glaucoma over the last really 5 to 10 years, but especially actually really over the last 5 years. And how, we are just beginning now to develop the tools where we can use genetics clinically to identify people who are at high risk for severe disease. And this, of course, is a great advance and a great opportunity to improve the care of patients with glaucoma. These are some not really conflicts with today, but some financial disclosures. So I think probably you all are very familiar with the disease glaucoma, and I don't need to belabor this, but just to make the point, of course, that glaucoma is a disease. First of all, it's the most common cause of irreversible blindness throughout the world. And the disease is caused, of course, by degeneration of the optic nerve, which we see here in this fundus image as an enlarged cup. But the degeneration of the nerve is related to the pressure in the eye, but the relationship between intraocular pressure and optic nerve disease is actually quite complex. We know there are people who have high pressure and never develop damage to the nerve, and then there are some people who develop damage to the nerve at even what we would say populationally are normal pressures in the eye. So our challenge in taking care of patients with this disease is to be able to identify people at early stages of disease, or even before they have any early stage of disease, when we can intervene with even, our current treatment for the disease, which is actually currently focused primarily on lowering intraocular pressure. But the problem is finding people who actually, are going to be likely to develop progressive disease, who are going to have severe disease, and how can we identify those people in an efficient way, and given the relatively common but relatively low prevalence of the disease in large populations. For example, intraocular pressure screening is not a good test because, as I just said, there are people with high pressure that never develop damage to the nerve. And additionally, doing things like optic nerve imaging, you really can't do at a population scale. So how does genetics help us with this problem? Well, to start with, interestingly, glaucoma, of prevalent human conditions, glaucoma is one of the most heritable of all human diseases. So what this means is that, genetics and specific genes have a major role in pathogenesis of this disease. So we can use those genes to develop genetic tests that identify people potentially who have high disease risk. And ideally, we can use those genes to detect people with high disease risk at early stages when our current conventional therapy is most effective. Another aspect of glaucoma genetics that I'm not going to talk about too much today, but is also very important, is that identifying the genes and understanding what the protein products of those genes do can also help us understand better the underlying mechanisms responsible for disease, and also, therefore, identify potentially new targets for gene-based therapies. So when we think about glaucoma genetics, it's important to understand that genetics affects people across the lifespan. And so there are early onset forms of glaucoma, like childhood glaucoma, juvenile onset primary open-angle glaucoma, the developmental glaucomas, congenital glaucomas. These are relatively rare in populations, but they affect children and babies. They are inherited as autosomal dominant, autosomal recessive traits. These types of glaucomas are caused by also mutations that are relatively rare in populations, but they have very, very large biological effects. And because of those large biological effects, these mutations, when they're passed down through generations, they cause disease. And that's why we find these kinds of early onset conditions in families. Traditionally, kind of in the whole beginning of the glaucoma genetics field, which was about 30 years ago now, we used these families for linkage analysis, sort of traditional linkage analysis. And now more recently, we've been expanding our search in familial glaucoma to using exome and whole genome sequencing. So currently we have 12 genes that are known to cause these sort of primary forms of early onset glaucoma, which is a relatively small number when you think about the number of genes that cause things like inherited retinal degenerations. We'll touch on that point more in a couple slides. So in contrast to the early onset glaucomas, there are adult onset forms of glaucoma, and these are the kinds of glaucoma that you're probably most familiar with. These are primary open-angle glaucoma, angle closure, exfoliation glaucoma. These types of glaucoma are relatively common in populations, and they are not caused by single gene mutations. These types of glaucoma exhibit complex inheritance, which means that they are the culmination of multiple genetic variants that are equally common, but individually have small biological effects. In aggregate, however, if you have enough of these common susceptibility variants, then you can reach a disease threshold and develop disease. Because it's of this sort of aggregation effect, we don't really see these diseases transmit in families as much as we find them in sort of sporadic or isolated cases, and then, of course, absent in controls. And so for this kind of gene discovery for these common complex diseases, we do genome-wide association studies. Currently, we have found more than 100 associated genetic variants for some of these conditions, in particular for primary open-angle glaucoma. Our most recent consortium paper identified 300 variants that predispose to this condition. So I'm going to talk about both of these today because genetic testing is really important, and I think we're making a lot of progress in both of these general areas, early onset and adult onset. So first of all, there are a number of benefits of genetic testing in early onset glaucoma. And these, of course, are families. And in these families, sometimes it's not clear among children in the family who actually is a carrier of a mutation, or if there is a disease mutation that's segregating in the family. So doing genetic testing with the genes that we know currently can cause these diseases, we can target surveillance, clinical surveillance in the clinic, and potentially therapy to people who actually are mutation carriers. This has tremendous benefits. First of all, by testing a family, we can identify mutation carriers before the disease becomes manifest. So for some of the, say, juvenile onset patients, they may not develop disease until age 10, 11, 12. And if we know that they're a mutation carrier, we can follow them very carefully and identify when they first develop disease and when they first need treatment. I think a really important point to keep in mind here is that we can also eliminate unnecessary surveillance of people in the family without the mutations. And so this is really a very important opportunity to not only reassure people in the family who don't have mutations, but also to simplify the lives of these families rather than having to bring every child in for repeat examinations to look for early disease stage. Another important point here is that we can inform genetic counseling and risk assessment in these families. These diseases, as I mentioned, were inherited as autosomal dominant and recessive, and so we can determine the inheritance model based on the mutation that they have and be able to give them good information about risk. Also important is that some of these genes have systemic comorbidities. Obvious example is PAX6 with Wilms tumor, but there are others that are important to know about. And in the future, when we do have some hopefully gene-based therapies that can target some of these early onset glaucoma genes, we can identify people who will benefit from those therapies. So those are all the benefits. Unfortunately, we do have some limitations for testing for patients with childhood glaucoma, and the most challenging of those is that currently with our current collection of genes, if we look at a population of 100 early onset glaucoma patients, only 20% of them are going to have mutations in the genes that we currently know. So that suggests that we have a lot of gene discovery to do, or at least a lot of genetics discovery to do. So we originally approached this problem of new gene discovery by targeting ethnically diverse populations. And the reason why we did this is because the genes that we currently know about have primarily been identified in European populations without a lot of ethnic diversity. And so we reasoned that we might be missing some genes and mutations that are perhaps more common in other types of populations. So we started with a collection of families from the Philippines that a really talented postdoc at the time, Ryan Calantes, had collected through, he is Filipino, and he recruited 12 very large, reasonably large pedigrees from the Philippines with early onset glaucoma. For these families, we did whole exome sequencing, so looking at all the potential coding sequences and variation that we can find in the coding space in these genomes. And then we first analyzed the data for mutations in known genes, and we found that indeed we found in 4 families, 4 of the 12, we found mutations in genes we already knew about. 3 of them had mycelium mutations and 1 had a PAX6 mutation that was diagnosed as early onset glaucoma. So then we moved on to filtering the remaining families for looking for the kinds of characteristics that we would expect in mutations causing early onset glaucoma, so rare mutations that are highly evolutionarily conserved, therefore have, likely a big impact on protein structure and function. And these are generally those that have predicted pathogenicity using our in silico tests as well. So then the variants that were interesting, we then looked for segregation in Ryan's families, and surprisingly, we found that 3 of his families segregated variants in a gene called EFEMP1 that some of you may be familiar with as a cause of some retinal disorders. So these are the families that we looked at that have the EFEMP1 variants, and you can see 2 of them are exceedingly large. 1 is quite small, but the segregation of these variants in these very large families really provides in itself, a sense of proof that these variants are truly underlying disease in these families. But we did investigate further. We found that all 3 of the variants that we found in these families were highly evolutionarily conserved, suggesting that they do have an important role in the protein. So as I said, some of you may be familiar with this EFEMP1. It's also called fibulin-3, and this is a causative gene, interestingly, for Doyne's honeycomb retinal dystrophy. There's a single missense allele that has been known for decades, the arginine 345 tryptophan, that is responsible in many, many different families, unrelated families, for the Doyne honeycomb retinal dystrophy. Also, about a decade ago or so, a US family, a primary open-angle glaucoma family, smallish family with a rare EFEMP1 variant, arginine 140 tryptophan, was also described. And then in GWAS, we have also identified association in the genomic region that contains EFEMP1 with elevated intraocular pressure in primary open-angle glaucoma. But this was the first report of EFEMP1 mutations in early onset glaucoma. So to get a little bit additional information about what might be going on here, we did some cellular assays, and we transfected cells with vectors containing these mutations. And one thing that we found is that the early onset glaucoma mutations, these 3 that we had found in the Filipino families, caused increased intracellular retention of the protein compared to both the variant that was found in primary open-angle glaucoma and also the variant that was found in Doyne's honeycomb dystrophy. So this has suggested sort of a genotype-phenotype correlation for EFEMP1, where the wild-type protein is normally secreted, and it's a normal protein that goes out into the extracellular matrix and does whatever it does, where the juvenile onset mutations have minimal secretion. We see no extracellular protein or subretinal deposits in these patients, and these mutations tend to keep the protein in the cell, limiting its travel to the extracellular matrix. Interestingly, we also tested the other 2 variants that have been discovered. The POAG variant, the arginine 140, has sort of an intermediate phenotype between the wild type and juvenile, where there's some sort of reduced secretion. And in some of these patients, we have identified some abnormal subretinal deposits. So some of the protein might be secreted, but then once it's secreted, it forms these deposits. And then, of course, the Doyne's honeycomb dystrophy, we think, is kind of a special case. It's really just this one amino acid where it seems to be secreted normally, but it's not a normal protein. And once it is secreted, it does form these subretinal deposits in the extracellular space. So this approach of looking in sort of diverse pedigrees was was interesting. And so we expanded this, this kind of study to look at some additional families and found another interesting story around thrombospondin. And here we found 3 different families, admittedly much smaller than the Filipino families, that had a missense allele affecting an arginine residue, arginine at 1034. And so 2 families had an arginine to a cysteine, a European Caucasian family, and then also an Indian mixed race family. And then another family from Africa had a serine at this arginine position. And the Indian mixed race and the African families were both identified by our colleagues, our Australian colleagues, Jamie Craig and Owen Siggs. So genetically, this is a really interesting finding where we have these 3 families of really very different ethnic origins with congenital glaucoma. I'm not sure I said that. These are congenital glaucoma families who have these missense alleles affecting this same amino acid residue in thrombospondin, of course, a very well-studied, very well-known protein. So we were really fascinated by this. The 1034 amino acid residue is extremely conserved through all kinds of evolution, but also thrombospondin 2, thrombospondin 3 in humans. So we decided, given the sort of small pedigrees and that we really couldn't put a lot of emphasis on segregation in these families, that we would try to make an animal model with this mutation. And we collaborated with the D'Amato lab at Children's Hospital in Boston and a very talented postdoc at the time, Haji Fu, he's now an instructor there, used CRISPR to introduce the missense change at 1034 into the mouse, and then proceeded to characterize the mouse model, which showed elevated intraocular pressure, pretty significantly reduced aqueous outflow, and also damage to retinal ganglion cells. But I think what was really an incredible finding that none of us particularly anticipated is that in the mouse, the mutant thrombospondin, which is the same mutation as is in the humans, causes this protein aggregation in the juxtacanalicular meshwork just adjacent to Schlemm's canal. So the big black arrow there is pointing out the staining of the thrombospondin, which is shown here in green. And you can see, compared to the wild type, just this really dramatic accumulation of this misfolded protein in this juxtacanalicular meshwork. We also showed that this accumulated protein increases over time. So in the panel below there, 1 month, 2 month, 4 month, the amount of protein is increasing. And using some electron microscopy, Fu showed that in the mutant, there are these dense fibrogranular deposits that form in the juxtacanalicular meshwork, and that the thrombospondin protein localizes with the aggregated protein in this mutant. So really interesting. We wanted to investigate further this sort of concept of protein misfolding here. And so Fu developed an in vitro assay where he transfected COS7 cells with plasmids carrying the mutations of interest, and then looked at the accumulation of extracellular material after the cells had been transfected. And I think you can see here in the panel A that in the mutation, the cysteine 1034, there's really a tremendous accumulation of extracellular material compared to the wild type. So we hypothesized that this was due to protein misfolding. And so we developed an allelic series. We tested an allelic series that had a range of protein misfolding effects, and showed that the amount of protein that accumulated is highly correlated with the impact on protein folding caused by these amino acid changes. So really interesting for further exploration to understand, what else is going on here in terms of how the thrombosbondin is behaving in the trabecular meshwork. One other thing that we've done over the past year, and Fu's going to be presenting this at ARVO for more details, but we've identified 3 additional families that have congenital glaucoma and also show evidence of this extracellular protein accumulation using this in vitro assay. And so one of these is another Indian family, actually a family from India with the same mutation as the prior ones that we studied in the mouse. And then 2 additional mutations, one in a US family contributed by Terry Young, and then another Filipino family that had another mutation. So we're really interested in understanding more about how these variants are impacting protein function. We have sort of a general hypothesis that these mutations destabilize the protein, causing this protein aggregation that then disrupts the normal function of the juxtacanalicular trabecular meshwork. But of course, there's much to learn specifically about the mechanisms here. One thing we are also really excited about, though, is that these 2 new genes, the EFEMP1 and the thrombospondin, these are both extracellular matrix proteins. And this, doubles the number of early onset glaucoma genes that function in the extracellular matrix. Most of the other genes that we have identified so far for early onset glaucoma are developmental in function. So they lead to abnormal development of ocular structures that are involved in draining fluid. And so very interesting in terms of clinical diagnosis and so forth, but perhaps mechanistically not quite as exciting as understanding how some of these proteins are working in the trabecular meshwork to lead to dysfunction and increase susceptibility to disease. So we're also, really excited about how complex the extracellular matrix is. And obviously this provides a lot of additional opportunity for study to really determine how these proteins are involved in disease development. So one thing that was perhaps a tiny bit discouraging from this study of Filipinos is that at the end of the day, when we looked at all the Filipino families and then compared, sort of the percent yield or percent solving with mutations, known mutations now, to what we had before, it really wasn't that great of an improvement. We still only find mutations in about 20% of these families. And so what's going on? So, we have some missing genetics, and I think there are, sort of 3 kind of general categories of where, these genetic effects could be, and that we're working on all 3 of these. And so the first is that, when we do genetic testing, many variants kind of get tossed aside. They maybe are relevant to disease, but, we kind of call these variants of uncertain significance because there isn't enough evidence or enough criterion to say for sure this is the gene of interest. And so to tackle this problem for all genetic diseases, the ClinGen consortium was formed about a decade ago now. And ClinGen is charged with really developing careful annotation tools for all inherited disease genes. Of course, there are many, many, many. But together with Rob Huffnagel, I lead the ClinGen ocular domain working group, which is now tackling all the inherited eye diseases, including those for glaucoma. So through this kind of effort, we're hoping that some of those variants that we toss away and don't get counted in our yield of testing, we might be able to restore with more appropriate rules for demonstrating pathogenicity. So another big topic is non-coding variation. And so, what we've really looked at so far is exome sequencing and looking at the coding space. So what about things that, aren't in the coding space that can cause, these large effect kinds of mutations like copy number variants and splice variants and things like that. So we've initiated a project now in collaboration with our Australian colleagues, Jamie Craig and Owen Siggs, to do whole genome sequencing on a large number of these families to see if we uncover some additional pathology. And then finally, and I'm going to talk a lot actually for the rest of the talk about polygenic risk scores, but so we can revisit this concept at the end of the talk. But it could be that what we're looking at for some of these patients is not actually true monogenic effects, but an extreme aggregate of polygenic effects that result in earlier onset and more severe disease. And so that's something else that we want to explore in our whole genome sequencing project. So I'm going to switch now for the rest of the talk to talk about adult onset diseases. And actually here, I'm going to talk primarily about primary open angle glaucoma. There's a lot of exciting stuff going on in angle closure glaucoma and in exfoliation glaucoma, but we don't have time to do everything today. So we'll concentrate on POEG. And I just want to remind you here that these, unlike everything I just talked about, where there are these big effects, where you have all this protein aggregation and all these problems because of these mutations, here, these individual variants that contribute to disease risk have small incremental effects. And so, as I said, in aggregate, however, disease, thresholds can be reached. But the challenge here is finding these variants that have small individual effects, because you don't find them by looking at them in aggregate, you find them individually doing genome-wide association studies. So how do you design studies with sufficient power that you can find these variants with little tiny effects? Well, you form consortiums. So, because no single group in the world had enough patients or cases and controls that you have a large enough sample size, that you have enough statistical power that you can show that a single genetic variant is associated with disease. So the first thing that we did, and we did this now, like 10 years ago already, we formed the Neighborhood Consortium. And this was among, sites throughout the United States that had collected cases and controls for genetic studies. Most of these were actually already genotyped. We did some genotyping as part of this. And included in the consortium, and one thing that really made this project so much fun is that we had ophthalmologists, for sort of the clinical aspects, but we also had, geneticists who knew how to design a genome-wide association study, and then bioinformaticists who could really help us with the analysis and identify the genes of interest. And so for the Neighborhood Consortium, we initially identified about 4,000 cases and 20,000 controls or something like that. And we were really fortunate to carry out this analysis that identified 3, at the time, quite important genes that contribute to disease. And at this point, it brought our total number of genes or total number of loci up to around, I think it was something like 20. This was around 2016. And but this still represented a fairly small percent of the heritability that I showed you way back in the beginning of this talk. And so we still were missing a lot, and we needed even bigger sample sizes than this. So we worked together with our international colleagues to form the International Glaucoma Genetics Consortium. And shown here on all these boxes are the names of all the data sets that were aggregated together to form a single sample that we could use for this, very large meta-analysis approach for gene discovery. And so we had samples from North America, but also from Europe, UK, and then Asia and Australia. So together we ended up with quite close to 40,000 cases and many thousand controls. This study still was largely driven by Europeans, but we were able to include a significant number of Asians, and also at the time, probably the largest collection of Africans. This was a couple of years ago. And we carried out then a genome-wide association study which looks at the distributions of SNPs between cases and controls. So these were our results from that meta-analysis. We found 127 different primary open-angle glaucoma loci, which are distributed. If you're not familiar with these, this is the so-called Manhattan plot. And what you're looking at here is the chromosome location of each individual SNP that we looked at, from the very top of chromosome 1 all the way through to the bottom of chromosome X, which is labeled here as chromosome 23. And then the negative log of the p-value for each individual SNP is shown on the y-axis. And that red line that travels across the bottom there, that represents the bone Ferrari corrected statistically significant p-value for this kind of study. You're looking at 600,000 SNPs here, 600,000 variants, and so that's a very large penalty for multiple corrections. And the accepted genome-wide significance for this kind of study is like 5 × 10⁻⁸. But despite all that, we found 127 primary open-angle glaucoma genomic loci, which was a very, very large increase a couple of years ago. I should say that recently, just in the last month or so, another study from our consortium has come out now identifying 300 loci using some additional meta-analytical techniques. So, progress is ongoing. So a couple of things that we were able to do with this study because of the varied ethnicity that we had in the data set is that we could compare the biological effect, the effect sizes or SNP effect sizes among different ethnic groups. And surprisingly, we found that actually there was reasonably good correlation between the European group and the Africans and the Asians. I mean, there are clearly outliers here, but perhaps more correlation in effect sizes than we would have originally thought, I think, going into the study. We also looked to see, kind of comparing to prior clinical trait GWASs and things, how the genes overlapped with other studies that have been found using cup-to-disc ratio as a variable or intraocular pressure as a variable. And we found that many of the genes that we discovered here were also involved in intraocular pressure regulation, but some also in cup-to-disc ratio. Some didn't seem to be associated with either one, and some were associated with both. We also did in this study a pathway analysis, just looking to see how the sort of nearest gene to the lead SNP correlated or was contributed to different biological pathways. And I think there are some that are interesting here. This is really just kind of data for future studies, but laminar fluid shear stress is clearly something that's of interest. There's a lot here with lipids and cholesterol, and then also blood vessel morphogenesis and vascular development. So those are interesting topics. But what we want to talk about for the next couple of minutes is how can we use this information now for risk stratification, or what information can we draw on this to be able to apply it to detecting high-risk individuals in populations, determining whether or not people are likely to progress to advanced disease, and can we detect people at earlier stages of disease. So as I had mentioned earlier, unlike early onset disease, where a single variant is sufficient to tip the balance from health to disease, for adult onset disease, each individual variant has a small incremental effect on disease, and only in aggregate do you reach that disease threshold. So it doesn't make sense to test an individual variant in adult onset disease. You really need to have a measure of this aggregate effect by testing multiple variants, a panel, some array that gives you an opportunity to say they are an extreme genetic burden to other people. So recently, in the last couple of years, the approach of using polygenic risk scores has emerged in human genetics. And this is not, only glaucoma, obviously. I mean, this was first become very popular in cardiovascular disease, neurodegenerations, cancers, breast cancer in particular, where the opportunity, if there is a sufficiently powered genome-wide association study like our recent one identifying 107 loci, basically the approach is to sum the risk variance from those identified in the GWAS for every person in a particular population, and then array in that population the distribution of the number of variants. So you have, obviously most people have an average number of variants and they're in the 50th percentile. You have some people who are at very, very high percentiles. They're the highest possible genetic burden. They're in the 90th percentile of distribution. And then you have people who are, the sort of lowest number of risk variants. And the hypothesis here is that people in the highest percentiles of this SNP, of this genetic risk variant distribution, those people in the 90th percentile and above, that they have the highest risk of developing disease, or they have the highest risk of having disease. Where the people in the lowest percentiles, the 10th percentile and below, those are people who are very unlikely to develop glaucoma. We don't know where people in the middle lie at this point, but that's our hypothesis. So to investigate this further, we created, and I should say that Ayelet Segre, whose picture somehow got deleted from this slide, but Ayelet Segre worked with us on developing a polygenic risk score based on this genome-wide association study. And we then applied the polygenic risk score to a population, a very large population in the UK Biobank. This is, a population of convenience. It's about 500,000, Europeans mostly, with some people in that population who also have glaucoma. And what we found is that when we stratified the UK Biobank according to the lowest deciles and up to the highest deciles, we found that people in the top decile had 7.7% of those people had glaucoma compared to 1.4% of people in the lowest decile, representing an increase in enrichment of about 5-fold in people with the highest polygenic risk. We also looked at clinical features that were associated with people with high polygenic risk and found that these glaucoma-related clinical features are also higher in people with high polygenic risks, especially intraocular pressure and corneal resistance factor. A couple of years prior to this, using a different kind of score, Jamie Craig had shown in his ANSRAG collection of glaucoma cases that among people with glaucoma, so not in a population now, but among people with glaucoma, those people in the top deciles had an earlier age of diagnosis, 5 to 10 years earlier than people in the lower deciles. Also, people in the higher deciles had retinal nerve fiber layer thinning and also had a greater need for surgical treatment for glaucoma, particularly trabeculectomy. We found in the UK Biobank also that people with glaucoma and a high polygenic risk score had a much thinner retinal nerve fiber layer thickness than people who were in the lower PRS deciles. Also, other studies that have been done recently, Owen Siggs, also part of Jamie Craig's group, showed in a different Australian cohort that people with higher polygenic risk scores also were more likely to progress with glaucoma. So these people were more likely to not respond appropriately to medications and actually showed progressive deterioration on visual field test. And recently, this study that was led by Nazli Zeberdash and our other colleagues at Mass Pioneer showed that in the ocular hypertensive treatment study, which is a study of people with high intraocular pressure followed now for like 20 years for conversion to primary open-angle glaucoma or optic nerve disease, we showed that people who were in the high polygenic decile, in this case tertiles, were more likely to convert to primary open-angle glaucoma than people who were in the lower distribution of the polygenic risk score. So, so just to kind of summarize that part, people who have high polygenic risk are more likely to have glaucoma. They're more likely to have severe glaucoma with higher pressure and more likely to have to progress and require surgical intervention for treatment. And, in this study, even among people who have high intraocular pressure, people who have uniformly high intraocular pressure, but also have high polygenic risk scores are more likely to develop glaucoma than people with high intraocular pressure and low polygenic risk scores. So that gives us a lot of opportunity potentially to look at this in the clinic. Now, a couple of other things about polygenic risk, which are kind of interesting. Polygenic risk scores can also modify other things. Like in a study that we did actually a few years back now, we showed that people who drank high amounts of caffeine didn't develop elevated intraocular pressure and glaucoma unless they also had a high polygenic risk score. So there was a combined interactive effect between high polygenic risk and caffeine intake. And then another study carried out by Nazli Zeberdash in our group showed that high polygenic risk can also modify the effects of patients with some of these early onset glaucoma genes. So in this case, she showed that people who have mycilin mutations, the people who have mycilin mutations and high polygenic risk had more severe disease than people who had just the mycilin mutations. And then another kind of interesting approach that we're investigating now with polygenic risk scores is sort of this partitioned pathway effect with polygenic scores, where we can look at a polygenic risk score for just a particular pathway. And here, Anas Abu-Baker, who's in our group, also looked at a polygenic risk score comprised of variants in TXNRD2 and MB3, which are two genes that are involved in NADPH metabolism. And she showed that these people with high genetic burden in this pathway are more likely to develop these paracentral or close to fixation visual field defects. But what's kind of exciting about this is that there's a lot of interest in NAD right now in terms of its protective effects potentially in glaucoma. There's actually several clinical trials ongoing looking at NAD supplementation. But NAD is a precursor to this pathway. And it may be that the people who are going to respond potentially the best to NAD are the people who have a high genetic burden in this pathway. So I wanted to just finish by telling you about kind of a real world PRS experience that we're involved in right now. And this is really, I think, a very important step towards implementing this polygenic risk score as a clinical tool in the glaucoma clinic. And so a few years ago, Lou Pasquale and I wrote an NIH grant to look at polygenic risk in our two representative biobanks at both of our institutions. So I used the Mass General Brigham Biobank, which is about 30,000 people, and he used the Mount Sinai Biomed Biobank, which is also about 30,000 people. And what we did in this study is we calculated polygenic risk for both biobanks, and then looked at the people who were in the very, very top and the very, very bottom in a clinical exam. So because of these biobanks at our institutions, we have an opportunity to bring these people into the clinic and actually examine them and find out really what they have. And so we're interested in sort of the clinical features associated with high and low polygenic risk, but also just what is the prevalence? What is the experience? I mean, how useful is this potentially as a test in the clinic? And we're going to present more data at ARVO on this, but what we have found kind of preliminarily is that people who are in the top 1% are 12 times more likely to have primary open-angle glaucoma than people who are in the bottom 1%. And that's a clinically important result. And one thing that I think is really, really interesting is that among the people that we've identified glaucoma in, when we've called them in, half of them have been undiagnosed. So these are people who didn't even know they had glaucoma. And through this polygenic risk testing and clinical exam, we have identified disease in these individuals. So this gives us hope that, you know, we can use polygenic risk profiling to tackle this problem of populations and how we find people who are at high genetic risk. And so, you know, through polygenic risk stratification, we can identify people at high risk, people in the middle, and people at low risk. And it may be that the people who are at high risk, those are the people where we really want to invest our dollars, our medical care dollars for screening, for surveillance. We can really target these people. We know that they're at high risk for disease. We know that they're likely to progress. We know that they will benefit from early treatment, and that we, you know, our focus can really be on them. We also, I think, can offer a real opportunity to people at the very bottom. These are people who are highly unlikely to develop disease, even if they have elevated intraocular pressure, as we showed with the ocular hypertensive treatment study. And so here, you know, being able to reduce healthcare dollars, reduce surveillance is also, I think, a tremendous benefit and something that can be very useful in the clinic. So just to summarize then, in summary, you know, genetic testing is important to identify carriers of high impact mutations, you know, allowing for surveillance and early treatment for early onset glaucoma patients. Using this, you know, sort of geographically diverse approach, we've identified 2 new early onset glaucoma genes, the FEMP1 and thrombospondin. You know, we have obviously a lot more to do in the early onset glaucoma space. Also, genome-wide association studies have identified hundreds of primary open-angle glaucoma risk variants, and polygenic risk scores can be used to identify people at high disease risk and with more severe disease with earlier onset. We think that pre-symptomatic polygenic risk stratification could allow for increased surveillance and earlier treatment, which would be of such benefit in the clinic. Also, interestingly, the background polygenic risk score can modify other risk factors, both genetic and environmental, and then this kind of approach of partition PRS may be useful for identifying, you know, kind of subgroups of interest within this sort of, you know, very heterogeneous collection of POAG patients. And I didn't talk about it at all today, but gene-based therapies for glaucoma are emerging and hopefully will be emerging more in the future. So lots and lots of work to do. It's such an exciting time for our field because it took so many years to really develop the resources that we need here. And now I feel like in the last 5 years, we've really kind of reached where we need to be in terms of resources and methodologies. But we still really need a better understanding of the genetics of early onset glaucoma. I mean, I think there are more genes to be found. I think we need to do a better job of figuring out what mutations actually cause disease, which we're doing with the ClinGen. We need to look at these non-coding effects with whole genome sequencing. And then I think there is a real opportunity here to consider polygenic effects as one factor that contributes to early onset disease. And then for POAG, you know, I think we're getting close to a point where we want to think about rolling this out in the clinic. But I think we still have questions that we would like to know. You know, are there specific clinical features that we find in these high, you know, PRS people that can potentially identify them, you know, even beyond the genetics? Also, are there factors that modify PRS risk? I showed you how PRS can modify other things, but are there other things that modify PRS? In other words, are there people who have high PRS that should have disease and they don't? And are there people who have disease and have low PRS? And why is that? And then the kind of familial transmission of PRS is something that's not well known in the field, and something that we also need to consider for glaucoma patients. So I will stop there. I'd like to acknowledge the neighborhood people, the original neighborhood consortium, which have just an incredible group of people to work with. And then equally incredible are the people in the International Glaucoma Genetics Consortium. Puya and Stuart McGregor in particular led that 127 paper that I showed you, and Jamie Craig has contributed so much to the glaucoma analysis in this consortium. And then these are all the people that work with us on different projects, not all of them that I talked about today, but in particular, I want to note Ayelet Segre and Nazli Zeberdash in our group at Mass Eye and Ear, who have done so much for the polygenic risk analysis that we've carried out. So thank you very much.
Yeah, as always, it was so clear. Thank you very much. We have several questions, so let me start with the simplest possible. Is the physiology of mouse and human in terms of glaucoma studying very similar, or there's huge difference between those two species?
I think I missed the very first part of that. Sorry. Is the what? I'm sorry.
Is the mouse and human, how similar the physiology, the system is?
Oh, for glaucoma.
Yeah, for glaucoma.
Yeah, that's a really good point. So one of the things that I think we and others in the field have really struggled with is intraocular pressure in the mouse. So, there are two pathways for fluid to get out of the eye. There's the sort of traditional pathway through the trabecular meshwork and Schlemm's canal, and then there's the uveal scleral pathway, which is, sort of the less traditional alternative pathway. And it turns out that mice abundantly use the alternative pathways. So if you have a protein that's targeting sort of the trabecular meshwork, Schlemm's canal, it doesn't impact the pressure as much as you might find perhaps in humans or other animals. And in fact, when we looked to see where else in the mouse the thrombospondin was accumulating in the mutant mouse, and we found that it was not accumulating in the tissues involved in the uveal scleral pathway. And interestingly, we found it not in very many other places in the entire mouse. So we didn't find it in other organs like kidney or pancreas or the heart, or even significantly in other parts of the eye.
All right, we have other questions here.
Thank you for that really interesting talk. So I was kind of curious, are there any human data to suggest that local epigenetic or metabolic changes in the retina or in the eye in general contribute to the development of adult onset glaucoma? And are there complications with that due to the lack of primary donor tissue to study, to do like epigenetic or metabolic profiling?
Yeah. Great question. I mean, it's really a developing field, the epigenetics in glaucoma. There are some studies that have been done in some animal models suggesting that there are some epigenetic effects related to aging that might be relevant to glaucoma. We haven't found anything through our genetic work that would point to specific epigenetic changes. But it is quite, an interesting field in development, and I'm sure there will be, further study in that area in the future.
Thank you very much for a very comprehensive and fascinating talk. So I have a question about the THBS1 story. So could you elaborate on the location of the amino acid 1034 in the 3D model? Is it like located outer surface or embedded to other amino acids? And why does the substitution with the glycine has the largest aggregation in the data?
Those are good questions. And actually, I would refer you to a lot of the details to our paper that was published in 2022. It's in Journal of Clinical Investigation. So the 1034 amino acid is in a fairly conserved domain in the carboxy terminal of the protein, and that domain has a lot of functional features. It's involved in binding fibronectin, it's involved in TGF-beta. It has, a number of possible biological, roles. But what we did find is that the effect of the missense alleles in the mouse, we looked at all of that in the mouse, and we didn't find abnormalities in those particular pathways or processes. But we did find that these, this is an important amino acid residue in terms of stabilizing the protein. And so there's more details about that in the paper. And one of the new mutations that we've identified actually is in a part of the protein that interacts with the 1034 domain in the protein as well. So there may be, some intraprotein structure that is important for maintaining, the role of thrombospondin in the extracellular matrix. But there is a lot more work to do to understand the precise mechanism, I think, underlying those mutations.
Hi, Jenny. This is Gulab. I thoroughly enjoyed your talk. I have 3 questions. It was nice to see all of the genes you looked. They tend to cause aggregation in the TM. So based on this, when you are talking about gene-based therapies, because these fibrillin 1 or thrombospondin may have a function, normal function also, would you develop more knockout strategy or changing the mutations? And the second question is that since these genes are like myelin, are expressed throughout the body, and actually these one are actually plays some known roles in normal physiology, what is that why we see this effect only in most in the TM?
Great questions. Great questions. So first of all, all the proteins that we've seen, that we've identified so far, are secreted into the extracellular matrix. And so, mycilin follows this pattern, as you noted, thrombospondin, and also EFEMP1. And then there is this, protein misfolding phenomenon, that has different impact. I mean, for EFEMP1, it seems to keep it primarily inside the cell. Mycilin, it seems to keep it primarily inside the cell. Thrombospondin, the protein secreted, but then it aggregates extracellularly. I think that if you think about gene-based therapy for all of those approaches, I don't think you would necessarily consider a knockout. I think, as it has been shown for mycilin, being able to delete, well, I guess, I guess in terms of knockout is, if you're thinking about reducing expression of the protein through a knockout, then yes, that would be an approach that you could entertain for all of these potentially. I mean, certainly that worked with mycilin, where Val Sheffield showed in 2017 using CRISPR that if he reduced expression of mycilin in a transgenic mouse model, that the intraocular pressure went down in that model. And I think that's an important preclinical study, to consider when we think about therapy for patients with mycilin mutations. I think something like that potentially could also work, I guess, for EFEMP1 and thrombospondin, but unlike mycilin, where I think we have a clearer view of the mechanism, I think still we have unanswered questions around the mechanism of EFEMP1 and thrombospondin 1, because we don't know exactly what the binding partners are there, what potentially we're doing to the extracellular matrix, and whether loss of those proteins could actually also be detrimental. So we have a bit more work to do in terms of thinking about how to approach therapeutics for those. Maybe a lot more work to do. So
Thank you for that.
Sorry. Oh, sorry. I see that Rachel has her hand up.
Yeah, hi, Janey. Always enjoy your talk. That was just fantastic presentation. So the fascinating, I think I have two questions. So some kind of, they are both related. You showed that the OATS data using the polygenic risk score, the conversion from ocular hypertension to POAG almost mirrors the IOP conversion, with or without treatment for those two groups. I wonder that if you could elaborate. I suspect you have quite a bit of data already. If you just like an old study, if you put central corneal thickness and IOP together, you have some kind of exponential prediction power. Do you have data using the other like IOP, central corneal thickness, the polygenic risk score in certain population? You can predict higher power, higher accuracy of a conversion, or the higher risk of patients needing aggressive treatment. Like similarly, for the congenital glaucoma, early onset glaucoma, early study like a myocilin, you have a myocilin mutation that can cause, you have a higher risk for even just POAG patients. Are we down the road, if we were to test those patients, are we going to incorporate everything, those 12 genes with the polygenic risk score and the clinical features, higher, up to this ratio, higher IOP? What is the future, for the clinicians? What are we going to?
What is the question you have?
The question is that if you can predict, being a clinician, seeing the patient, what test, you can do to predict the patient with a higher chance to get a glaucoma?
Yeah, well, that's a great question. So going to the OATS first, the OATS study is actually relatively on genetic scale small. So the power to show some of the relationships that you identify was more challenging. And also one thing that I didn't emphasize in this talk, but is also in our paper that just came out in JAMA Ophthalmology is, OATS is roughly a third non-Caucasian, non-European. And so, that also potentially limits, the power that we have there. So I can't really give you an answer to, how much the PRS is contributing to risk prediction in addition to those other clinical features that we find. And it's something actually that we do want to look at more in the clinic as we continue to study people in the clinic who have high polygenic risk versus low polygenic risk, and what clinical features we find associated with that. So, and some of those risk alleles do impact intraocular pressure. So that's another consideration when we're looking at a population like that. With OATS, I would like to think that the difference between the people who convert and the people who don't, with regard to polygenic risk, that's emphasizing the role of genes that might influence optic nerve degeneration primarily over intraocular pressure. But, that's speculation at this point. With regard to your second part B of your question, I actually think that given the results that we have with how polygenic risk can influence, say, myocilin, and it doesn't influence every myocilin mutation necessarily. The one in that study is the most common one that is also the mildest one, which might be the one that is most likely to be influenced by polygenic risk. But nevertheless, I think it's kind of hard to give clinical outcome information to patients with myocilin mutations without having a polygenic risk score. And so actually for that reason, we're starting a new study in the clinic where we're rolling out a test that we're going to offer to patients it's a research basis, but we're going to carry it out in a CLIA lab so we can give them information. And we're going to be testing for early onset glaucoma genes and polygenic risk score, so that, if somebody does have like a mycilin mutation, or even say a FOXY1 mutation or LTBP2 or something else, we can, also look to see if they have a high polygenic risk score as well. And I think that will be I think it's interesting on a research basis, but I also think it will be important in terms of clinical information that we can offer to patients.
So we still have a lot of questions, just to let you know.
Okay.
Let's take with
I know, well, Don had his hand up
with
for a
the
while.
Zoom. So Don.
Hi, Janey.
Hi, Don.
As everybody's already said, that was a wonderful presentation and summary of 5+ exciting years of work. So it's really cool. So I have two PRS kind of related questions. One is, as you sort of already alluding to the role of PRS in the future, as of right now in your clinical practice, how often do you ask a patient to get genetic testing, and how often does it make you maybe treat a patient a little more aggressively or not? Is this all still in the future, or is it actually affecting clinical care in 2024, would you say?
Yeah, so in 2024, 23andMe offers polygenic risk score testing for primary open-angle glaucoma. And it's interesting to me that not that many patients show up with their numbers, but some of them do. And so far, I have not seen anyone show up with a high polygenic risk score from 23andMe. But what the information that 23andMe gives to patients is, as you might imagine, very limited. And I think that we just, I would not feel comfortable necessarily acting on, something where I didn't know more about the patient at this point. So that's actually, though, one of the reasons why we are starting the study that we are studying, because, I want to see really how does it impact our care. So when we start, what we want to do is, everybody who comes through the door, we want to do this genetic test. We're not going to charge them for it. We're going to do it as a research test, as I said, because I don't think it's right to charge people for this. I think it's too early. But I think from doing that, we're going to learn how the patient responds to the info, and perhaps more importantly, how the doctor responds to the info.
Right.
And that's
you made the comment
goal.
of not using it without knowing more about the patient, but I think the implication is it's still you're a patient that you know well. Question is, will this, there is a subjective decision, as you obviously know very well, how aggressively to treat, whether that affects things. But and a related secondary question, with the different genes and variants that go into the PRS, is there any good evidence of synergy between different genes? Is that providing any insight that some variants are much more active when in the presence of another variant?
Well, that's something we would love to know, and we don't know the answer to that. That's a whole nother frontier. Probably would take a much larger data set than what we have so far, even in, like, say, the UK Biobank. The partition PRS thing that I talked about just a tiny bit, that begins to sort of scratch the surface of that. Because if you find that you can look at, say, all the variants in genes that are clustered within a pathway, and then look at perhaps, how that pathway interacts with clinical features in patients, that might begin to give you some insight into how those genes are potentially interacting. But that's a very, very early days for that kind of study. And it you can imagine that if you start subdividing this into smaller and smaller subgroups, that
Sure.
you're going to lose power. So, but
All
yeah,
right,
I think
let's move to Valerie.
Okay.
Hi, thank you for the great talk. My question is kind of following Dawn, so thinking about clinical practice as well, but do you think it would make sense, or do you see the use of genotype criteria or that PRS risk score being utilized for glaucoma neuroprotection or neural enhancement trials from a clinical trial perspective? So for example, identifying patients that could be enrolled in those trials?
Yes. Yes, I think that's a really important consideration. Absolutely. And actually, some of that is happening in other diseases, you know, in cancer and also in cardiovascular disease. They are incorporating risk stratification as part of the selection criterion for people that are enrolled in those clinical trials. And I think it would be a really interesting opportunity, you know, to select individuals who are perhaps more likely to progress rapidly and would, you know, have a shorter duration of outcome, say for neuroprotection. So yeah, I do think that's important.
Andrew.
Thank you for this great talk. So in residency, we learn about multiple theoretical mechanisms for glaucoma development, vascular versus mechanical. And I was wondering if in the 127 to 300 genes, if they support one of these theories more in terms of prevalence than another, in making hypotheses based on what the gene's roles are in their topographic expression levels, and which of these theories would be more likely to be most prevalent.
I'm sorry, something came up on my screen in the middle of your question and I missed part of it. Do you mind saying the question again? I'm sorry.
Sure, no problem. So in residency, we learn about different theories for the evolution of glaucoma, vascular versus mechanical, and others. And I was wondering if the 127 to 300 genes you've identified in terms of their topographical expression within the eye have a higher likelihood of supporting one of these theories more than the other.
Yeah, good, really good question. You know, we find collagen genes, we find vascular genes. I think they're both right. I mean, I don't think we have anything to refute one versus the other. I think that, you know, and I think too, we have to remember that glaucoma, primary opening of glaucoma is genetically quite heterogeneous, you know, and I think wouldn't be surprising that there are different ways to get to the same kind of common endpoint. And perhaps even more so when you think about things like normal tension versus high tension glaucoma. And we haven't done a lot of that yet. So we are working hard on a normal tension glaucoma GWAS, which is, you know, just a larger scale GWAS for normal tension glaucoma, because a lot of that has been missing from current studies. A lot of the cohorts that have been involved in our current GWASs have intraocular pressure as part of the selection, you know, criterion for a case. And so they naturally would have more intraocular pressure genes. So we're trying to really focus on those people that are so-called normal tension, and see what, you know, maybe we find additional genetic susceptibility variants there, and maybe adding those to the PRS is also going to be useful. So, you know, we have that approach to look at too, but I can't help you with your residency problem, because I think they're both I think they're still very valid hypotheses.
Dr. Wiggs, this is Dominic. Great talk. I have a question. When showing your mouse model, you mentioned lipid metabolism and lipid deposits. What is your opinion on the association between hyperlipidemia and basically lipid metabolism or metabolic health of an individual and the glaucoma risk or the glaucoma development?
You know, I think that's a great question. There are a number of sort of lipid cholesterol related genes that show up in these different, you know, pathway analyses that we do and things like that. I'm not really sure what that means. I think one thing that we have focused on, you know, maybe not as much currently, but in the past, is some of these, you know, for example, like the caveolins, CAV1 and CAV2. That's a genomic locus for primary open-angle glaucoma, and also ABCA1, which interacts with those genes. It's also involved in lipid metabolism. And some of those, you know, that pathway in particular is involved in membrane physiology. And I think there's some concept around the importance of, you know, sort of normal membranes, intravehicular meshwork, you know, to function for, you know, the intraocular pressure egress. it may involve these giant vacuole things that have been described for decades, and they're still not super well understood, but there are these big sort of vacuoles that pinch off from the membrane and trabecular meshwork, especially in the juxtacanalicular meshwork near Schlemm's canal. And, membrane, disrupting normal membrane function may have something to do with that. But I think there's, again, that's kind of speculating.
Thank you.
Hi, Jenny. This is Dorota Skowronska-Krawczyk. I actually am very interested to hear your thoughts about the intersection of glaucoma and aging. So you mentioned that some group of risk factors, risk variants have earlier onset and so on, but your work is like kind of looking for every risk factor, like genetics for every patient. Am I right or not? And then if not, what group of patients you would expect doesn't have particular risk factor related to glaucoma? It's mostly because of the lifestyle, I don't know, some other exposures.
Well, that's great. Yeah, age is an important risk factor for glaucoma, and we have shown that there's an age effect even at this paper isn't published yet, but we've looked at age effects across the PRS spectrum, and we do find important age effects interacting with PRS. And so I think that there is still obviously an important contribution of age with regard to to glaucoma development. I think one thing that is really interesting to think about, and we originally were really excited about this, but in our kind of real world study where we have this distribution of high and low PRS patients, we were interested in seeing what's wrong with the people in the low PRS that develop glaucoma. Are these people that have, say, a myosin mutation that had not been detected, or are these people that have some other environmental influence that might cause glaucoma, or even aging in that instance? And so one of the things, we've had very few people in the low percentiles that have glaucoma. So we don't really have enough people yet that we can address that. But I do think that there is the opportunity in those people who have low PRS that do develop glaucoma to determine some of these non-genetic, or at least non-glaucoma genetic causes. They may be, genetics of some other disease, but that may be contributing to pathogenesis. So, I think that's an interesting opportunity, and one that we haven't yet been able to explore ourselves in our current data sets.
So this was truly super informative. We will finish with two questions, one from web and one from here. Tony, you're the first to go.
Thank you very much, Janey. I was sort of interested in the slide in which you talked about paracentral scotoma and high genetic burden associated with that. Can you explain that more to me?
Sure. Yeah, and that paper is also in Ophthalmology in, I think, 2021. But there were two genes that we had high confidence in from the GWAS that are involved in mitochondrial function. So these are not encoded by mitochondrial DNA, they're encoded by nuclear DNA, and they're TXNRD2 and ME3. TXNRD2 is a protein that actually is involved in the thyroxine detoxification, free radical removal in mitochondria. And then ME3 is a protein that actually produces NADPH. So what's really interesting, though, is that what we found is that the effects of these two genes were in opposite directions, and it's because TXNRD2 consumes NADPH while ME3 produces it. And so when we aligned the risk in the right direction and then looked at the genetic burden of variants just in those two genes, then we looked at their clinical features and found this, this interesting association with paracentral scotomas, which is the kind of defect that you can find in, people with mitochondrial dysfunction, because there's, of course, a high density of mitochondria in that, paracentral region. So, Yeah, so that's what that is about. And because of the relationship also between NADPH and NAD, we think it's interesting because there are these, studies looking now at NAD supplementation as a possible neuroprotection or neuroenhancing treatment for glaucoma. And so it'd be really interesting to know, if the people who respond best, if anybody responds, but if the people who respond best to NAD are the people who have excess burden in this particular pathway. So but the paper might describe it a little bit better and in more detail. It's in ophthalmology.
Thank you.
Yeah.
Thank you for your really interesting talk. So you've shown a lot of these glaucoma risk variants, but do you think your data sets are powered enough to look for glaucoma protective or negatively correlated variants in your patients?
Right. Well, so protection, okay, yeah, a lot to say here, and I don't have a lot of time, but so risk and protection in sort of the complex scale of things is relative. So, if somebody has increased risk, then the flip of that is that somebody, with less risk variance might be felt to be protected in some way. But to actually look at real protective variants, which I think is probably what you're getting at, there haven't been that many of those identified in glaucoma patients. There has been one, and it's been super interesting. I didn't talk about it today, but angiopoietin-like 7 variants, it's also an extracellular matrix protein, and missense variants in that protein are protective. So people that have these missense variants, unlike, EFEMP1 or thrombus bondin or myocillin, where the missense variant causes increased risk, these individuals have decreased risk. And so we don't understand a lot yet, really, about how that works. We do know it's an extracellular matrix protein. We do know that the mutations cause the protein to be retained intracellularly. And actually, Dan Stamer just published a really nice study on angiopoietin-7 and trabecular meshwork, and I think it's in IOVS, if you want to learn more about that. But it's a really, it's a really interesting topic. And the mutations in this gene are relatively rare. So we would need a very large data set, to be able to see these effects, but you can see them in data sets, say, the size of the UK Biobank, which is around 500,000 individuals, or, the original paper that described these took advantage of enrichment of some of these variants in the Finnish population and showed in FinGen, which is a kind of a biobank of Finland, that they also are protective. So, we thought maybe in our, we might be able to observe an effect of some angiopoietin-like 1 variants in our top 1% who should have glaucoma and don't. But so far, we haven't been able to look at that yet. So maybe we will, and maybe we'll be able to find other potential protective variants or protective effects by looking at that category of person in the top 1% who should have disease and they don't. So that would be, an interesting group for that kind of discovery.
All right, please join me to thank Dr. Wiggs.